Happy Friday, everybody. Hopefully you survived the cold. I know it was a painful week for me. I had a couple days of my kids being home because the buses weren't running, and then I had 3 days of my kids needing to get picked up because they had final exams because they're in high school now. Not quite old enough to drive. It's been busy, hectic, chaotic. But I'm recording this at 1:45 because that's when people are quiet. So, this is our next paper, Huntington's disease, and sort of jumping forward a decade almost, right? And the sort of techniques or technologies that might be available. Look through 6 or 7 different groups worth of answers. It looks like you're honing in on some stuff, right? You're like, some of you looked up what the enzyme cuts, things like that. But some of you still, be hitting the target on some questions and maybe not quite hitting it on others. We're learning, right? Make sure I'm on the right screen. So start off. I've got sort of who's working in your group, who published this paper, how is it important. Talk about that one even though I don't have it on here. So probably when you read a paper, now, in the future, whenever it might be, there's 1 to 10 authors. There's not a whole ton of authors. Usually it's from 1 or 2 different institutions. Science is collaborative, but science is also quite specialized, right? In that people sort of have their gene or their disease or their organism or their organelle or whatever it might be that they're studying. And in this case, this paper is published by the Huntington's Collaborative Research Group, and with a little asterisk on it saying that, it's not the name of a person, that's the name of the group. And it's because they have so many authors on this paper, so many people whose work was critical that they were like, they deserve authorship on this paper. And they realized the way that the paper system allows for citations and stuff, whose name goes first. Well, we've got multiple labs who have done this work, and all of them have been important. None of this could be done where if you'd sort of taken away one of these. So this paper has 58 authors from 6 different research groups. You've got ones in Boston, you've got ones in California, you've got London, you've got Wales, you've got California, all over the place, all English-speaking, sort of places, but, a couple different countries, different hospitals, different universities and research centers, and, it's because this has been a disease that people have been trying to sort of figure out for a decade in this case. If you were to look at the list of names, the most famous person on this list is in group 5. He's the last author down here, and it is Francis Collins. About 5-6 years later, Francis Collins became the head or the lead of the Human Genome Project, which led to, in the early 2000s, the human genome being fully sequenced and something sort of a benchmark that we could compare all of our genomes to see if we have mutations and things like that. And he's been director of the National Institutes of Health. So sort of big scientist, Fauci level, sort of, running the show type of thing, but at the NIH, not the National Institute of Allergy and Infectious Disease, NIAID. Got a cat scratching on my chair right now. Go away. I don't know. Get back to the topic at hand. So, who published this paper? A really wide collaborative group. Why is it important? Because you don't see a lot of papers with so many authors on it. What is Huntington's disease? A lot of you answered this well. There's sort of the background in the paper, and maybe you can just look up a easy Wikipedia article type of thing. But Huntington's disease is a dominant, neurodegenerative, fatal disease. So if we sort of think about these sort of parts, move my microphone a little bit. Dominant, fatal, neurodegenerative. I'm so lazy that I don't write and I don't think about letters and all that. Dominant. Why is dominant important? Dominant is important here because if you carry this gene, you will get the disease. There is no, as far as they understand at this point, there's no ambiguity. There's no, maybe you're a carrier and maybe you'll pass it on as a carrier to the next generation, and, you know, sort of like every once in a while it pops up like cystic fibrosis or something like that. Huntington's disease, if you have the mutation, you will end up getting the disease. And that means you have a 50% chance of passing it to your kids. We know that at the time, even back in 1983, when they sort of figured out what chromosome it was on, this is not a sex-linked, it's not X-linked or Y-linked, it's autosomal, which means both male and female children will have an equal chance of having this chromosome being inherited, the one that has the disease. And it's fatal, but it's fatal usually in the 40s and sort of more severe ones up to, you know, 70s. But the fatal part, or at least even the beginnings of exhibiting the phenotype of the disease, doesn't happen until most people are past reproductive age. And this is sort of one of the tragedies of Huntington's disease, in that you would have people who maybe would make a decision about whether they want to have kids or not based on whether they actually have inherited it. You could imagine that, you know, if you're someone who today was diagnosed and saying, yes, you have the mutation for Huntington's disease, you will guaranteed get that disease and you will die from it, you might say, I don't want to have a chance to pass that on to my kids, or maybe I don't even want to have kids because I don't want to leave them, you know, without a parent at some point in the future. I don't want to have them have to see me deteriorate, things like that. Like it's become a real sort of ethical dilemma. And even then they knew it was dominant and then it was fatal, and that nobody knew until after they've already passed it on. So there was this sort of, you know, impetus to, let's at least try and figure out a test for it so that we can let people know that they are going to have the disease so that they can make those decisions. The neurodegenerative nature is you got a movement disorder. Eventually you end up getting like emotional dysregulation, depression, and eventually it's fatal. But the main thing is sort of this Huntington's chorea, this like movement shaking sort of disorder. So in the 10 years previous to this paper being published in 1993, they have done the work to find out what chromosome this gene is on. So they did sort of pedigree type mapping. We're going to see some pedigrees, but you can imagine that you get a family that has multiple kids, and then you look to see which of the kids inherited that disease. So you need to have these long longitudinal studies, or you need to be looking at really grandparents and parents. You can't look at children yet. Much better to do if you have larger and larger families. If there's 10 kids or 15 kids, then you're better able to sort of say, well, the kids that got it, which chromosome of the parent did they inherit? Did they inherit this chromosome 1 or that chromosome 1? This chromosome 2 or that chromosome 2? And by looking at banding patterns and like, you know, karyotypes like this, they can start to narrow down and say, oh, you inherited this, you know, visible marker that gives us a phenotype, and you didn't inherit the disease, and it turns out they're both on chromosome 4, so maybe that helps us sort of narrow it down, right? So they figured out that Huntington's disease is linked to chromosome 4. That's one here. And then they start mapping it onto chromosome 4, saying that once we localize it to that chromosome, we can look for molecular markers like RFLPs, write that down for you, old technology, restriction, fragment, length, polymorphism. You can imagine that if you have a chunk of DNA and it could either have an EcoR1 site, or maybe there's a mutation that destroys that EcoR1 site. You know, one base could be changed and the enzyme won't cut anymore. A restriction, fragment, length polymorphism is essentially we cut up the DNA and do a southern blot, and the band changes size depending on whether you inherited this chromosome 4 or that chromosome 4. So they've got these markers here, and they've sort of been mapped onto this chromosome. They've been localized as to where they are on the chromosome, and they then map those and find out sometimes they get co-inherited with Huntington's disease. Sometimes there's a recombination event where it looks like the parent had this marker and Huntington's disease, but the child had the marker and no disease, or the disease and no marker, and that tells us that the distance between those on the chromosome was far enough that there was a recombination event, right? So we had, this has got the disease, this has got the cut site, this doesn't. Maybe sometimes we have a recombination event in there so that through meiosis, we inherit a chromosome that has both the disease and the restriction site. So mapping, pedigree, they start to narrow this region down. And they've gotten it down to the short arm, or the P arm, chromosome 4, so this 4p16.3. The numbers after the P or Q, in the case of the long arm versus the short arm of a chromosome, essentially, when they needed to map things visually to a chromosome, they looked at the banding pattern, and big bands got the big number, and then there were sort of sub-bands or smaller bands, smaller, darker areas that got like the decimal sort of numbers, and they've localized this to 4p16.3 and sort of narrowed it down to this region that I believe was 2.2 million base pairs. Oh, I have that written down somewhere. Did I just get off the top of my head? 2.2 megabases. 2,200,000 base pairs of DNA. This is a lot of DNA. This is, in the case of bacteria, half of an E. coli chromosome. Could have hundreds of bacterial genes in this. In the case of human genes, we could have hundreds of genes, or potentially we could just have one gene within this region. If you did take BMSC 320, the old Biochem 300 with me last term, or part of the term when I taught some of it, when it comes to prokaryotic genes, we have a lot of density. There's not a lot of space between the genes. We don't have introns that need to be spliced out. But with eukaryotic genes, we have these huge spaces in between genes, and the genes themselves, exons are typically quite small and introns are quite large, which means there's a lot of sort of empty space, or at least non-coding space in the genome. So this 2.2 million base pairs, there could be a single base potentially that is the critical factor that determines whether you have the disease or not. And there is not the technology in 1993 to just, let's sequence chromosome 4 in everyone with Huntington's disease and then run it through a computer and find out what was the mutation they all had in common. It's not possible at this point in time. They need to essentially narrow it down more, and sort of hopefully find the gene and find some other stuff at the same time. So the method that they're using for this 2 million base pairs is a process called Exon Amplification. And this is something that probably wouldn't be done nowadays, but at the time, it was the way to find a gene within a region. So they've taken chromosome 4 and they've broken it up into like yeast artificial chromosomes, or even like, 100,000, 200,000, 300,000 base pair sort of chunks. And they're like, well, let's start finding genes in these chunks. So you can imagine if we had a big chunk of DNA and potentially, let's imagine we've got a gene here, and a few exons, and maybe on the other strand, because it can be reading in the opposite direction, with its exons like this. Maybe over here, there's another gene, exon. So the boxes are what we're looking for, but this entire DNA is what we have sort of accessible to us. How, in the absence of sequencing this entire region, do you find out where the exons are? Well, splicing is a process where exons get stitched together after it's been read into messenger RNA, right? So the process of exon amplification went like this. We have a vector, and this is a eukaryotic vector. It has to be because it needs to be spliced. And we've got a promoter. We've got an original exon hooked up to that promoter. You can imagine that this is our 5' untranslated region. And then we've got a cloning site where we're going to be inserting our foreign DNA. And then we have another exon. So exon, maybe, stop codon. This isn't going to be translated, so it doesn't really matter if it encodes a protein or not. But if we were to take a chunk of this DNA, so this piece, or this piece, or this piece, or this piece, inserting into there a chunk of DNA, and a lot of it is non-coding intronic or intergenic space, but if there is one or more exons within this, and they are not the first or last exon, they are an internal exon that is going to have the splice sequence at both the 5' end to accept the splice from the previous exon, and at the 3' end of that exon, a splice donor to then, allow it to splice to the next exon's 5' end, right? So you can imagine that we are going to make a messenger RNA like that, right? And it's going to have exon, 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 and then it will get spliced to remove this, remove this, remove this, and we will essentially get box, box, box. All right, so these two are the ones that have been inserted, and this is the first, this is the last. How do you determine if an exon has been put in there? It is by doing RT-PCR. So we've got a messenger RNA that's been spliced. We are going to reverse transcribe it to DNA, and then we are going to do PCR from the first to the last exon. And the size of that PCR product, if there's no additional exons inserted, is going to be our sort of base size. Maybe it's 100, maybe it's 200 base pairs, but it's all going to be the same size regardless of what inserted fragment if there are no exons in there. If there are exons present, that is going to increase the distance between those two PCR primers. And when we amplify it, the longer product will tell us that, hey, we trapped an exon. We don't know what that exon is. We don't know what gene it might come from. All we know is that something got spliced in there. It's interesting. Let's make a molecular probe from that and figure out where it actually maps to on the chromosome. Let's maybe sequence it and, start to use maybe that as a molecular marker or something like that. But we found that there is a gene present in that region. So they are doing this exon trapping. Do I have it written down? So they are doing 500,000 base pair fragments at a time that they've sort of broken that 2 million into, and then chunks of that 500,000 get used for exon trapping. Once they find an exon, this is going to be a probe that's used for a cDNA library. So we can make a cDNA library from a Huntington's disease individual or from a normal individual, probably from brain tissue, expecting that this is a neurological disorder, a neurological disease. So we're probably going to have expression of the Huntington's disease gene in the brain. And we have no idea what the sequence is, but if we've got an exon that's been trapped, that can make a DNA probe that goes to sort of fish out the rest of the messenger RNA as a cDNA in our cDNA library. So cDNA library is essentially every messenger RNA from a tissue, from those cells, has been converted into, reverse transcribed into DNA, cloned into vectors, put into essentially tens of thousands of clones on plates, and each clone might be a completely different cDNA. We don't know which one it is, but once we have a probe, we can use that to find like the individual colonies that contain a cDNA that has an overlapping sequence. So they do this with this 2 million, and they start working from one end, they start working from the other, and they have found a few genes here that we found a gene. Great. Let's sequence it. Let's run it against the pedigrees. Turns out that this gene has nothing to do with Huntington's disease. It's just some other gene that was in this region. They search again, they search again, they search again. Eventually, they are going to find this interesting transcript 15, as it's called. Check my notes. Make sure I'm on the right page. So they've been searching for over a decade, going over exon trapping. They have put a restriction map on here for us. And this restriction map, sort of how do we organize these big chunks of DNA when we have pieces that are so huge? Like this is our 500,000 base pairs, our 500,000 right here. You can't use an enzyme like SAW3A that they did for that TAC library last time, or the partial digest. It's a 4 base pair cutter. It's going to cut really often. They are using NOT1 and MLU1 that are both 8 base pair cutters. I think I've got that right there is where they list the two enzymes, NOT1 and MLU1. Both 8 base pair cutters, which cut approximately every 65,000 base pairs. So these are going to be long interval restriction fragments, which means they're going to be larger in size. So they've sort of mapped this out, and they've got these like chunks here, and essentially they're, trying to find the cDNA, trying to find the gene. This is right here probes that were discovered through exon trapping. There's those sort of stars there. And they use these to fish out cDNAs from the cDNA library. Problem, especially back then with doing cDNA libraries, is you might have an RNA that's poly A tail. We can enrich or isolate the polyadenylated RNA from the rest of the RNA, like ribosomal or tRNA from a cell, by essentially binding it to a oligo DT sort of cellulose or beads, or passing it through a column, where essentially the TA pairing retains the messenger RNAs and the other RNAs flow through. And then we can use a DNA probe that is essentially run of T's, 5' end, 3' end, and use a reverse transcriptase to copy back from that. So essentially we copy this into DNA. We might not copy all the way to the 5' end of the messenger RNA, the 3' end of the cDNA, which means we might get a fragment of a cDNA instead of the whole thing. And also, we might initiate the reverse transcription not at the poly A tail, but maybe there is a slightly homologous run of adenine somewhere within that gene, and that's where we start. So sometimes we could get a cDNA that is truncated on the 3' end or truncated on the 5' end. We aren't necessarily going to find cDNAs that are the whole thing. So they use these probes and they sort of pull out a few different cDNAs, and through molecular biology sort of analysis, they figure out these are actually all stitched together. They're part of the same cDNA, and they come up with this sort of composite interesting transcript 15, which is eventually going to be called Huntington or Huntington's disease gene. If this is a gene that is expressed in the brain, now that we have a probe, we should be able to do a northern blot. So isolate RNA, run it out on a gel, use this probe to try and figure out the full length of the transcript. And they found out, like, here is, here is our gel, 18S and 28S ribosomal RNAs for sort of sizing. And we can say, we've got this big transcript up here, and this transcript happens to be like expressed in all these tissues. But essentially, we've found something here that is being expressed in the brain. So next they say, okay, we've got a transcript, just as these other ones that they did that turned out not to be the right thing. this ADDA, this ID10C, this IT11. And then they're like, okay, we've got a gene, it's expressed in the brain. Let's look in diseased and non-diseased individuals to see if there's any differences, right? And what they found when they did the sequencing of the genomic region, which was this normal cosmid. Cosmid was a sort of semi-viral large vector that contains large inserts, but essentially a library fragment. The normal cDNA, so a normally expressed, not the DNA copy, but the RNA copy, and a Huntington's disease, so someone who had the disease, they also made a library from them, and they started to do this sequencing and comparison using old-fashioned radioactive sequencing gels where they had to, like, each lane was an individual letter because it would have been like, don't need to explain how the sequencing worked back then, but dideoxy sequencing where the 3' end of the last nucleotide would not be, it would be not have a hydroxyl, which means we can't add one more base to the end of it. And essentially we would sort of synthesize DNA until we hit this like premature stop where we couldn't add any more. And that terminated that fragment of DNA, and it also labeled it so it'd be radioactive. And as these ran through the gel, we would start off with the smallest fragments to the largest fragments, and each time we see a band, it would terminate in an A or G or C or T. You can sort of see you've got our 4 lanes labeled out there. Later on, they went to fluorescent sequencing where each of the 4 bases had a different fluorescent color, and then it can run in 1 lane instead of 4 lanes, and then we can automate it, and we can have like laser scanning to figure out sequencing. And you were going to learn from Hong about like next generation sequencing, things like that. But this was like old-fashioned dideoxy radioactive sequencing. Each lane is radioactive with a different sort of terminal nucleotide. But one of the weird things that they found when they were looking at the sequencing is they had this repeated pattern of like, there's an A, and there's a G, and then there's a C, and then there's another A, and then another G, and another C. A, G, C, A, G, C, A, G, C, A, G, C. It looks like we've got this trinucleotide repeat, right? And it turns out that it starts at the C here, A, G. So C, A, C, A, G, C, A, G, on and on. Found in both the normal DNA copy, the normal RNA copy of the cDNA, and the Huntington's disease copy. But it looked like that latter extended longer in the diseased individuals. So it looked like this has some promise. This might be the actual change that's found in these diseased individuals, is that maybe there's this extension of these trinucleotide repeats. So once they sort of narrowed down to this, like, it looks like this repeat is longer in diseased individuals and shorter in non-diseased individuals, they said, well, maybe that's just one individual. It could be that it just happened to be that in that one individual with Huntington's disease, they had this mutation in a completely different gene, and that's what we're chasing down. Like, there's no guarantees yet, right? So what they then go to is the pedigrees. We can see shorter triplet repeats in the normal copy, longer triplet repeat in the disease copy. Does this trend continue for every diseased individual? And basic pedigree, mom, dad, whole bunch of kids. They mark them all off as triangles because they're like, we don't care whether they're male or female, because essentially this is not a sex-linked thing. We are not looking at sort of multiple generations where we're like, you need to make sure a boy and a girl are paired up. But what we've got here is the mom was completely normal. She didn't have Huntington's disease, and she had 2 different lengths of that CAG repeat, but they were both short. The father had bands here and here, not that visible. Papers back then This is sort of right when they started scanning and putting stuff online in the next couple of years, and the first papers they scanned were really bad resolution. So sadly, the sort of images in this aren't that great. But let's sort of see in an afflicted individual, we've got the large band. In a non-afflicted, we don't have a large band. Afflicted individual, large bands. Unafflicted, small. And it looks like, in this pedigree at least, there is this trend where if you have the big one, you get the disease. If you have the small one, you don't. And looking at the sort of range here, they found that in the chromosomes of people who had Huntington's disease, 30 of them had between 42 and 47 repeats. 44 of them had more than 48 repeats. Never on a Huntington's disease chromosome did we have less than 24 repeats. 169 times on a normal chromosome that didn't cause the disease, we had less than 24 repeats. So it looks like there's this good sort of concordance between repeat length and having the disease or not, right? And then they're sort of like, is there a critical number, or is it sort of like a dimmer switch where, this range is normal, this range is maybe a little diseased, and it sort of gets worse as you go along? Or is it sort of like there is this critical threshold where below, you're fine, you hit the threshold, you are going to get the disease, and above that, you're going to get the disease. We're trying to sort of tease this out a little bit more. So they keep looking at pedigrees, not just this one, looking at others. Are there any sort of, can we find a single instance even where a diseased individual doesn't have the longer copy? And, don't really need to go through the multiple pedigrees they have, but essentially it's always the case that the long copies get the disease, and if you don't have the long copy, you don't. They do some of this with PCR as well, and I think they even find a couple individuals where the size of the expansion is so long compared to a normal individual. You can imagine all these additional repeats as opposed to a few repeats, that it actually blew out their PCR, where the conditions that they had set up to amplify weren't set up to make a piece that long or amplify a fragment that long. They didn't give it a long enough extension time. They didn't sort of have well purified enough DNA to actually amplify it. But then they went and did the Southern blot again. And they're like, yeah, that person has the long copy. So they managed to sort of narrow this down and really sort of set out these rules that are still the rules today, where if you are 36, 37 repeats or less, you're not going to get Huntington's disease. If you have 37, 38, you are going to get the disease, but not till later in life. And they even find in this paper that they had some cases of juvenile Huntington's disease where they start being afflicted really young, and these are the individuals that have 100 or even 200 repeats, right? So what do they sort of know? Well, they've narrowed it down. They can figure out that we now have a genetic test for Huntington's disease, and we can do that at least sort of advisory role to people and say, we're going to test you. We can let you know if you are going to be an afflicted individual or not. We can inform you whether your kids are afflicted or not, whether you passed it on. You might know before you have reproduced, and maybe, that's going to change your decision on whether you're going to have kids or not. What do they not know? They still have no idea what this gene does. And something I didn't really sort of talk about is the question, I've got the questions on my other screen here. How do you find a disease gene when you don't know anything other than it exists? Paper last week when we were looking at Taq polymerase, we at least knew the enzymatic activity, so we could try to purify the enzymatic activity and use that to generate antibodies that were specific to the enzyme, right? Here, we don't know what the normal gene does or what the normal protein does, so there's no activity that we can track. And because it's dominant, this could be a completely new function for this protein that is not related to its normal function, which means we don't even know if this is like, oh, well, this is a disease in the brain. Maybe it's a neurotransmitter receptor, or maybe it's a axon growth regulator or a myelin thing, or who knows. No, it doesn't necessarily have to do anything with normal brain function. It can just be this gene, when broken, affects the brain because it happens to be expressed, but it has nothing to do with normal neurological function, right? So they don't know what this protein does. All they know is that these CAG expansions, this polyglutamine disease, which ends up that there's actually a class of, or a group of diseases that have similar phenotypes, where if you have this expansion that makes this sort of long repeated run of glutamines, these aggregate in the brain and cause the disease, and it has nothing to do with the normal function of the protein. So what do they know? They know how to track it. They know how to test for it. What do they not know? They don't know what the normal gene does. They don't really know why it causes a disease phenotype. And there is no sort of treatment indicated by this, right? It's not like they were like, oh, it turns out that this is an enzyme, and if we add this food back into your diet, that's going to reverse the disease. This is not one of those sort of simple ones that we can figure out. And, they are pretty optimistic in this paper, where they're like, great, now that we know what the disease is or what the gene is, we can come up with a treatment because we're so smart. 25, 27 years later, we still don't have an effective treatment for Huntington's disease. We still don't even really have a good idea of what the normal protein does, because the diseased copy, or the polyglutamine expansion copy, that is what people have really focused on, not what does the protein normally do, but why does that aggregate and actually sort of lead to toxicity in neurons. So 25 plus years later here, we know that it's the expanded protein that causes the damage. There's no pharmaceutical treatment. There's no way we can sort of change the gene regulation. But we do know that if you have 35 or less repeats, 35 or fewer, you are not going to live long enough to come up with a phenotype for Huntington's disease. 36 to 39 is a little bit of a gray area where, if you get hit by a car when you're 50, you'll probably never have exhibited any indication that you actually had that sort of premutation range. But if you live to be 90 or 100, you might start to see some neurological sort of breakdown, right? If you have 40 or more, definitely you're getting it. The higher you are, the earlier you're going to get the disease. And I'll leave you off with this paper from 2019. What year is it? 2021. 2020 happened, but it didn't, right? So a year and a half ago, about September 2019, CRISPR-Cas9 mediated genome editing increases lifespan and improves motor deficits in a Huntington's disease mouse model. So we are now, through the advent of CRISPR, being able to do things like, maybe if you've got 2 copies of the gene, and one we've got all these expansions and the other we don't, can we use CRISPR to knock out this copy while leaving this one functional? And that's the stuff that we had to develop completely new technologies in order to target a disease like this. This isn't one that we can do easy gene therapy for, where we're like, you aren't expressing this enzyme, let's get this enzyme expressed in your eye and suddenly your vision will come back, or express this enzyme in the liver and suddenly you don't need to have these, blood transfusions or whatever the case may be. This is not something so simple, because this is a gain of function. This is a new protein that's getting produced that's degrading neurons, and the only way to treat that is to lower the expression of that gene. So either by manipulating the gene expression, changing the splicing so the RNA doesn't get translated into protein, or using something like CRISPR, which could be a permanent fix in that individual's neuron if you can somehow target this DNA to either cut out or completely destroy the gene expression from the disease copy. So we're getting there. There's still no guarantee that we'll ever get all the way to a treatment, because you probably need to target every single neuron in the brain in order to get this to function, right? So what happens if we only get 10% of the neurons to sort of downregulate expression of Huntington? Maybe that does lead to some sort of improvements. Maybe when we try and use CRISPR and go from mice, which don't have a highly complicated brain function, to, testing this in primates or something like that, maybe it would turn out that, oh, yes, it reverses the disease, but it has some off-target effects and affects things, right? Like there's no guarantee just because it works in mice that it's going to end up working in human, but there's some promise now that we didn't have even 5 years ago. That's the end, I think. Thanks for watching. See you later.